First, you have your cell phones turned off or turned to vibrate. Our next speaker is presenting on genomic pedigree reconstruction provides novel insight into the reproductive ecology of the brown treesnake, Boiga irregularis on Guam. The speaker is Brenna Levine from the University of Arkansas, Pedigree.
Good afternoon, everyone. Can you guys hear me okay? So my name is Brenna Levine. I'm a graduate student at the University of Arkansas. And a primary goal of my research is to understand the mechanisms that link ecology and evolution on a contemporary timescale. And the reason that that interests me is that the management of wild populations, specifically those of invasive species, which we'll talk about in just a second here, can be very affected by slight changes to the relationship between their ecology and evolution. So let's talk about invasive species for a second. So invasive species are those that are introduced from a location out in their native range to a new location outside of their native range, that become established in that new location and then spread beyond that initial point of introduction. And invasive species are a devastating force for global diversity, as you guys are all aware, I'm sure. And one of the reasons that invasive species are so successful is because of their reproductive ecology. Their reproductive ecology facilitates their ability to become established in an environment if they're able to produce enough offspring to overcome a small propagule size. And it also allows their population to grow and continue to persist in the face of stochastic environmental change when they start out at small population sizes. And when our effort is on focusing on how to control invasive species, we can actually take advantage of the fact that our control mechanisms might end up impacting that relationship between ecological processes and contemporary evolution. And this can go the wrong way sometimes if, for instance, we inadvertently select against controllable phenotypes, which we'll talk about in a few minutes here. My specialty specifically is the brown tree snake. So the brown tree snake was introduced to Guam around 1949 as the US was salvaging residual military material after World War II in the Pacific. And after only a handful of animals were introduced in 1949, by the 1980s there were over 2 million brown tree snakes on Guam. And this is a very small island. You can drive the perimeter of it in a few hours. I've done it myself. And when you introduce a species like that and it grows so rapidly, it can cause some pretty devastating ecological and economic damages. So one of the most notable ecological changes is the decline of Guam's avifauna. So for instance, there are 12 species of birds that are no longer found on Guam, 3 of which were endemic. One of those is completely extinct. 2 of them are only found in captivity right now. We're hoping at some point we'll be able to reintroduce them. And so because they are so damaging to the ecology and also the economy when they get tangled in electrical wires and cause massive power outages, there's been a lot of effort on trying to figure out how best to control this species. And because birth rate is such a fundamental demographic parameter, a lot of that focus has been on understanding the mating and reproductive system of the brown tree snake. Unfortunately, brown tree snakes are notorious for being difficult to study in terms of their mating and reproduction in the wild. So to give you some context, this is arguably one of the most well-studied species of snake on the planet, and we have never observed it mating in the wild. Never happened. In the history of studying the brown tree snake, I mean, reproduction itself has been focused on since the 80s. Only about 3 or 4 clutches of eggs have ever been found. So it's kind of a really big black hole in terms of how we can control them. Now, fortunately, DNA can allow us to reconstruct familial relationships among individuals. And if we're able to look at a pedigree, that family tree, we can actually infer mating and reproduction patterns from that family tree. And so that's what the focus of my talk is going to be on here today. So the goal of this project was to reconstruct a multi-generational pedigree for a wild population of brown tree snakes on Guam so that we could identify predictors of mating and reproductive success in a natural population. So annual mating success is being defined here as the total number of mates with which an individual produces offspring during a year. Annual reproductive success is the total number of offspring that that individual produces during a year. And once we can identify those predictors of annual mating and reproductive success, we can then consider them in the context of existing control methods to determine whether or not our current control is effective at targeting highly fecund individuals, and to evaluate whether or not our current control is likely to remain effective over time, or if some of the traits involved in reproduction are in fact heritable. And we'll talk about that at the end of our talk here. So there were four main predictors, or four main variables that we evaluated in terms of whether or not they seem to influence annual mating success in brown tree snakes. These were the age of the snake. You can see my old snake, you know he's old because he's wearing glasses. Hey, I'm wearing glasses too, don't worry, they're just hidden in my eyes. The body condition of the snake, the personality of the snake in terms of whether it's a shy snake or a bold snake that's more willing to take risks. You know this one's shy because it's behind a tree. And how inbred the snake is. So we evaluated these four parameters because each one focuses on a different aspect of biology or ecology of the brown tree snake. We also hypothesized that those four parameters, in addition to a mating parameter, which is the number of mates with which an individual mates with, whether or not these five parameters would influence annual reproductive success, or the number of offspring that an individual has during a year. So our study site is a geographically closed population that is in northern Guam on Andersen Air Force Base. It's surrounded by a snake-proof fence, and it's 5 hectares large. And within the population, snakes just behave, you know, they have their normal day, right? So this population was closed in 2004, and since 2004, we've collected 424 DNA samples from this population, which is pretty close to a complete enumeration of the snakes that have lived in the population over time. And in addition to collecting DNA from these snakes, each snake has an extensive capture history where we have body mass over time, SBL over time, both of which have allowed us to determine estimated birth dates or birth years for each of the snakes. We have body condition over time, and importantly, we have records of how many times each individual has been captured in a baited trap over time. So throughout the course of this study, baited traps have been deployed to determine whether or not we could figure out why certain animals are entering traps and why some are never entering traps. And so we're going to be using that propensity to take that risk and enter that trap today as kind of a proxy for personality, that risk-taking behavior. So each sample was sequenced at thousands of loci within the genome using a reduced representation library preparation technique called DDRAD sequencing, double digest restriction site associated DNA sequencing. And this resulted in millions of raw sequencing reads for each individual, which were then clustered de novo into loci, resulting in 6,180 single nucleotide polymorphisms that were present each in greater than 95% of individuals in the population. And some of these loci were then used for pedigree reconstruction using an R package called Sequoia. So we chose the most informative loci for pedigree reconstruction. These were loci that were in low linkage disequilibrium, had no departures from Hardy-Weinberg equilibrium, and that had high minor allele frequencies. And the result here is what you can see is a multi-generational pedigree. So the oldest snake, or the earliest one, was born in 2002 in our population, and we have snakes all the way up to those that were born in 2016. And to give you an idea of what we're looking at, the red dots up here represent the birth of a female, the blue dots are birth of a male, blue lines are offspring of those males, and red lines are offspring of those females. And so from looking at this pedigree, we can then get an idea of the annual mating success and annual reproductive success for each one of these individuals over each year of this study. Once we had those estimates, we then constructed some generalized linear mixed effects models in order to determine which fixed and random effects affected our annual mating success and our annual reproductive success. So our first mixed effects model it predicted that it modeled annual mating success as a function of the fixed effects of the age of the animal, the body condition, how bold the animal was in terms of the number of captures that in a trap that it underwent, and its inbreeding coefficient. Annual reproductive success was modeled as a function of the age, body condition, boldness, inbreeding coefficient, and number of mates with which the individual produced offspring. And you'll notice that both of our response variables, the models also included 2 random effects to account for the dependency structure of our data, where we had repeated measures of many animals over multiple years. And because of the large time span that it spanned, we also included year as a random effect to capture some of that variation as well. And also, these generalized linear mixed models were run in a sex-specific way, so males were run alone, females were run alone, just so that we could remove that additional fixed effect and save that degree of freedom. So let's get to the fun stuff, which is the results. So 149 individuals in the population were assigned as offspring to 60 known females in our population, and that's what's represented by these red dots and red links over here. Whereas 235 individuals in the population were assigned as offspring of 50 known males. And these are known as our dams and our sires. So now that we know who reproduced in the population, we can then get estimates of our annual mating success and annual reproductive success for those dams and sires, those that actually reproduced. So on average, when a female reproduced, it mated with 1.5 males. When a male reproduced, it mated on average with 2 females. And this resulted in an average of 2.6 offspring per female per year, whereas males experienced greater reproductive success, but also greater variance in reproductive success, as you can see here. And this result alone was really exciting for us because even the concept of clutch size has eluded brown tree snakes for 30 years. And so this is kind of the closest that we've gotten so far, which is very exciting. So moving on to the results of our generalized linear mixed models, after model selection, we found that our top model for male annual reproductive success included significant partial effects of the age of the animal, the body condition of the animal, and how bold that animal was, how willing it was to take risks and enter the trap, a baited trap. Same went for females. We found significant partial effects of the age of the female, the body condition of the female, and the boldness of that female as well. On to male annual reproductive success, our top model identified partial significant effects of the age of the animal, the body condition, and the number of mates with which that animal was able to mate with. Females had similar results with a significant effect of the age and the number of mates of the female. So let's look at this in the context of why this is important. It all comes back to control with the brown tree snake. So upon first glance, these seem like really promising results if, say, we're zooming in on one of these traits, the boldness, the willingness to enter a baited trap. Baited traps are the main way on Guam that we're controlling brown tree snakes right now. And it's those snakes that are most fecund that are also the ones that are entering baited traps most often. This could be a good thing. If we continue to deploy these traps, remove individuals that are caught in them, over time, we may end up with a population with individuals with reduced reproductive success. But we need to proceed with caution in case boldness, a major axis of personality, also has a heritable genetic basis. So over time, we could end up with a population of snakes that are refractory to capture. So we have many people to thank for this project, including all of the snakes that participated in this project, including this one that let me stare at it. And I thank you all for coming today.